40 milliliters of hydrogen peroxide in a beaker, beaker number 1. Make sure hydrogen peroxide in beaker number 1 is 22 degrees Celsius or room temperature. Place the 40 milliliters of hydrogen peroxide into 200 milliliter of room temperature water. With forceps, hold a hole punch paper circle and catalase for 5 seconds. Place the paper circle on a paper towel for 5 seconds. For forceps, put in the paper circle at the bottom of the solution. Time how long it takes for a paper circle to rise to the surface. Record how long it took. Name that trial 1. As you can see in the graph, the oxygen production was most effective at 25 degrees Celsius or room temperature.
Based on our data, the colder the temperature, the longer it takes for the paper circle or substrate to rise to the surface. For example, when the temperature was 25 degrees Celsius or room temperature, it took 19 seconds for the substrate to rise. When the temperature of the hydrogen peroxide was 5 degrees Celsius, it took 36 seconds for the substrate to rise. And when the hydrogen peroxide was 75 degrees Celsius, it took 27 seconds for the substrate to rise. The enzyme in this case, which was the catalase, was controlled and did not change. The dependent variable was the oxygen production that lifts the substrate, and the independent variable was the temperature of the hydrogen peroxide, which we changed for each of the 5 trials. If we could change anything, we would have done the experiment in order, starting with room temperature at 25 degrees Celsius for trial 1, hot at 45 degrees Celsius for trial 2, hot at 75 degrees Celsius for trial 3, cold at 15 degrees Celsius for trial 4, and cold at 5 degrees Celsius for trial 5. This would have helped us keep track of our data better.
Bill Nye the Science Guy. Bill Nye the Science Guy. Bill Nye the Science Guy.
Science rules.
Bill Nye the Science Guy.
Inertia is a property of matter.
Bill